Good morning, students. Myself, Sapna Chaudhar. I am from IIMT College of Pharmacy, Greater Noida. Today, we are going to discuss about the unit first of the subject, medicinal chemistry in B Pharma fourth sem. So, there are various topics which are included in unit first. Like first topic is your physicochemical properties in relation to biological action, then history and scope of medicinal chemistry, and the next is drug metabolism, that is your phase 1 and phase 2 metabolism, and next is factors affecting drug metabolism. So first point included in this syllabus is drug physicochemical properties in relation to biological action. So there are various parameters which affect the biological action. First is solubility, then chelation, ionization, partition coefficient, protein binding. These are the factors which affect the biological action. So basically, medicinal chemistry is a discipline chemistry. This subject included the development of new drugs with their medicinal uses and the biological activity. So, the history, various scientists are discovered the medicinal uses of various drugs. And so first, here we are discuss about the physico-chemical properties. Then afterwards, we are going to discuss about the history and remaining part of the unit first. So first property which affect the biological action is solubility. So, the ability of a chemical compound to elicit pharmacological or therapeutic effect is related to the influence of various physical and chemical, that is, physicochemical properties of a substance on a biomolecule that interacts with. Physical properties of the drug is responsible for its action, and thereafter chemical properties are also responsible for the chemical action. So these are the parameters which affect the biological action. First is solubility, partition coefficient, dissociation constant, hydrogen bonding, ionization, redox potential, complexation, surface activity, protein binding, and isosterism. So, optical isomerism or geometrical isomerism is also included in this list. So, first one is solubility. Basically, solubility is just defined as the concentration of the solute particles in given amount of the solvent in the presence of in the presence of temperature, pH, or the other environmental factors are also included like pressure. So, like nature of the solute, it is hydrophilic or hydrophobic or lipophilic nature. So the solubility governs on that factors also. The atoms or a chemical compound or an organic compound are held together by various types of bonds in the solubility like hydrogen bond, dipole-dipole interaction. So there are various methods which we are going to apply for to increase the solubility like addition of co-solvents or alteration of pH, alteration of temperature. So, alteration of pH and temperature. The forces are involved in the solubility because of the solvent-solvent or solute-solute or solute-solvent interaction that govern the solubility. So these are the methods which are going to use for to increase the solubility. Structural modification by alteration of some changes or alteration of structure, we can increase the solubility of the substance. Then use of co-solvents, employing surfactants, which decreases the surface tension between the molecules and thereafter increases the solubility. And next is complexation. By formation of complex, it also increases the solubility. So how this solubility is important for the biological action? Firstly, bioavailability of drug is depends on the solubility of given compound in the particular solvent. So the drug like solute we can consider here must be in the solution before it is absorbed into the biological membrane. So and it shows its activity. So, and after the solution, it goes into the solution that binds or interacts with the various receptors. So, bioavailability is totally depend on the solubility. This is the biological importance of the solubility. Next, sorry. Next factor is protein binding. Simply, hydrogen bonding. Hydrogen bonding is simply a special dipole-dipole interaction between the hydrogen atom or in a polar bond, such as various electronegative atoms are present various electronegative atoms are present in the compound like oxygen or nitrogen or fluorine. These electronegative compounds pulls electron towards them. So the hydrogen bond, so the hydrogen is electron positive, and when any highly electronegative atom is attached with the hydrogen atom, the hydrogen becomes electron deficient, and they make a weak attraction and forces which makes the bond. Simply that bond is called as hydrogen bond. If that hydrogen bond is present between the molecules, it is simply called as intermolecular hydrogen bonding, and within the molecule, it's simply called as intramolecular hydrogen bonding. In case of hydrogen bonding, it is denoted by dotted line, and with the help of dotted line, we can indicate the presence of hydrogen bond on any compound. So what is the importance of the hydrogen bonding in biological action? Many physical properties are affected by the hydrogen bonding, like intramolecular hydrogen bonding generally decreases the melting point or the decreases the solubility. Also, in case of intermolecular hydrogen bonding, they generally increases the boiling point, melting point, or the solubility of the compound. And it is important for the gastric acid also, chemistry of gastric acid, and important for the receptor interaction as well as the biological activity. So for the stability, protein by hydrogen bonding is essential, and for the stabilization of hydrogen bond, it increases the it makes the bond within the molecule or between the molecule. Next is protein binding. Protein binding is simply the bond between various plasma proteins and the bond between various drugs. That bond formed between the plasma proteins, simply various proteins are available in the body like albumin, globulin, or any other albumin, globulin, or other glycoproteins are also present in the body. So if the bond between the plasma protein and the drug, that bond indicate the protein binding. So basically protein binding are irreversible binding also or the reversible binding also. In case of reversible binding, the protein and the drug complex is formed. That protein and drug complex is going to be sorry for the interruption. This is the example of hydrogen bonding. Hydrogen bonding, here is one example like orthonitrophenol. It is simply indicate the intramolecular hydrogen bonding. In orthonitrophenol structure, oxygen of the nitrogen atom is formed the bond of other hydroxyl group, and which is denoted by the dotted line. And in case of second example, intermolecular hydrogen bonding, two molecules of para-nitrophenol form the bond between the molecules, and simply it is also denoted by the dotted line. Next is next is protein binding. Protein binding is irreversible or reversible. In case of irreversible protein binding, the drug and the protein complex is formed, and that formation of that complex with the very weak attraction. In case of weak attraction, that bond is easily breakable and the drug is going for its activity. And after showing its activity, the bioavailability of the drug increases. In case of irreversible binding, the attraction between the drug and the protein is formation of covalent bond over there, and that covalent bond increases the attraction between the drug and the protein. So that drug is cannot reach to the site, so its bioavailability decreases. So what is the importance of the protein binding in biological action? It influences the bioavailability and distribution of the active compounds. Because of the irreversible binding of the drug, bioavailability decreases. Both complex and the free drug are important for the complete pharmacological action. Then next is partition coefficient. Partition coefficient is simply denoted by the K, and simply that partition coefficient is the ratio between the two phases at the equilibrium. That partition coefficient is indicated by the P, and it is denoted by the P. It indicates the lipophilicity of the drug or the nature of the drug. Either the drug is lipophilic or hydrophilic, which is indicated by the partition coefficient. Highly water soluble medication cannot permeate lipid rich organs such as brain or other neural tissues, whereas compounds that are particularly lipophilic will be held in the initial site of loss such as fat tissues and will be unable to leave the site rapidly to reach the target. The partition coefficient indicate the how drug is transported and distributed, as well as how they are going to reach to their targets. And the is the drug concentration equilibrium constant for two-phase molecule, because partition coefficient in living system are difficult to quantify. So these are next factor which affect the biological action is bioisosterism. In case of bioisosterism, simply the study of bioisosters are generally called as the bioisosterism. In medicinal chemistry, bioisosters are also a chemical compounds or the chemical substituents which have similar physical and chemical properties with similar biological activity. In case example like carbon dioxide or nitrous oxide, one scientist named as Friedman proposed a definition of bioisosterism, the phenomenon by which compounds usually fit with similar functional groups are simply called as isosters and possess the same biological activity. Simply, the phenomenon is called as bioisosterism. They are either classical form of bioisosters or non-classical bioisosters. Classical bioisosters are also divided into the monovalent, divalent, trivalent, tetravalent, or ring equivalent bioisosters. In case of monovalent classical bioisosterism, these are the few examples which indicate the monovalent, divalent, trivalent, tetravalent, and ring equivalent bioisosters. In case of ring equivalent bioisosters, benzene or thiophene ring, benzoxazole ring, and indole nucleus are the some few examples of ring equivalent bioisosters. In case of non-classical, these are the some structural example, like benzene ring is replaced by thiol ring. This is example of classical bioisoster, ring equivalent bioisoster. Then non-classical bioisosters are either cyclic versus non-cyclic or the functional groups. They do not have the same steric or electronic configuration, but like classical isosters, they do not have the same structure, but they have same properties, due to which they give same biological properties or the biological activities. So what is the importance of the bioisosterism in the biological action? Bioisosters are broadly used for the pharmaceutical sciences, and in drug designing, the purpose of exchanging any bioisoster for another is to enhance the desired biological and physical properties of the drug without changing their structure, help in the production of new drug, which is safe and more effective as compared to previous drug. So bioisoster plays important role in the biological activity of the drug. They have cyclic or non-cyclic or functional groups. Multiple ways from the classical bioisoster. Non-classical bioisosters are different from the classical bioisoster, but retain the focus on provide similar steric and electronic profile to the original functional group. So they show the similar biological activities as like previous original drug. Example, a chlorine group may often replaced by trifluoromethyl group or by cyano group, but depending on the particular molecule used for the substitution may result in the little change in activity or either increases or decreases affinity or efficacy depending on what factors are important for ligand binding to the target protein. Like exchangeable group. First example, in phenylephrine structure, the phenolic group in phenylephrine may be replaced by alkyl sulfonamide group. Some of the resulting compounds are showing agonistic activity, whereas the previous compounds are showing antagonistic activity. In case of cyclic and non-cyclic structure, estradiol is cyclic structure while diethylstilbestrol is non-cyclic structure, but they both show the similar biological estrogenic activity. So that's why they are called as bioisosteres. Next is ionization. Simply, the ionization represents the process by which atom or molecules acquire the negative or positive charge by gaining or losing the electrons. They resulting in changes in atom or molecule because of the ions present in that molecule. Like drug is in an ionized form or ionized form. In unionized form, it shows the lipophilic nature. In ionized form, it shows the hydrophilic nature, and the absorption is also depend on the ionized form of drug because the drug reaches into the blood shows the drug having hydrophilic nature reaches faster to the blood as compared to lipophilic nature because of the nature of the biological membrane. So these are the what is the relation of what is the relation of ionization with the biological action. Role of ionization in the biological action is play an important role in the pharmacokinetic. It involves the adsorption, distribution, metabolism, and excretion. A good balance of ionized or unionized form of the drug is better for the ADME. An ionized form of the drug shows lipophilic nature and ionized form of drug shows hydrophilic nature. Ionized form impact good water solubility, whereas unionized form shows lipophilic property. So these are the basic examples of ionization. Next is optical isomerism. Physicochemical properties of molecule are only depend on the functional group are present in the molecule, but also special arrangement of these groups that become especially crucial factor when the molecule is subjected to an asymmetric environment such as the human body. Optical isomerism and geometrical isomerism. Optical isomerisms are be defined as simply the compound that differ only in their ability to rotate plane polarized light. The differences in biological activity between optical isomers depend on their ability to react selectively at an asymmetric center in the biological system. Geometrical isomerism. The term geometrical isomerism indicate type of diisostereomer that occurs as a result of restricted rotation around the bond as an olefinic compound. Generally, geometric isomerism indicate the cis and trans form of the compound. Cis form is more stable than the trans form of the compound, and they are showing usually same biological activity, but their spatial arrangement of the atoms are different. That's why they are called as geometrical isomerism. However, the geometrical isomers may exhibit optical activity, that is, they are having capability to rotate the plane polarized light. The observed difference in biological activity of geometrical isomerism may be due to part of the differences in interatomic distance of group essential for elicitation of the response. The classic example is diethylstilbestrol, a drug that is synthesized a mimic natural estrogenic hormone, and estradiol, the trans isomer of diethylstilbestrol, has 14 times estrogenic activity of the cis compound. That's why the cis form is more stable than the trans form. Chelation. This is another parameter which govern the biological property as well as physicochemical properties. The compound that are obtained by donating electrons to metal with the formation of ring structure are simply called as chelates. The compound capable of forming ring structure. Most of the metals can form the chelates or complex, but the chelating property is restricted to atoms like nitrogen, sulfur, and oxygen, which are electron donating. The phenomenon of chelation is significantly involved in biological substance, biological system, and to some extent in explaining drug action. Example, penicillamine is effective antidote for the treatment of copper poisoning because it forms a water-soluble chelate with the copper and other metal ion. Generally, this chelation property is used for the treatment of poisoning because of the various chelates form or complex form. That complex does not easily penetrate the biological barriers. That's why chelation property is important for the biological action. So other remaining part we are going to discuss later, and that's it for today. Thank you so much.